So hello dear guys, today we'll discuss the tubular digestive system, histology of tubular digestive system. So we artificially subdivide the digestive tract into tubular digestive system and also liver and pancreas, distinct parts of digestive system. So today we'll concentrate our attention on tubular digestive system. So here I wanna explain you basic histological organization of digestive tract. So I hope you remember from respiratory tract, so that we have some typical linings which are present in digestive of any tract we have there. It's mucosa, which is itself composed of 3 layers. In digestive tract, it always follows these 3 layers. So you remember, in comparison to respiratory tract, so in mucosa we also had 3 layers, but not everywhere. Now we have in mucosa we have epithelium, lamina propria, and muscularis mucosa. In respiratory tract, muscularis mucosa sometimes absent. It starts only from the secondary bronchi. But here mucosa is saved in digestive tract. So let's return discussion of composition of this mucosa a little later. But now let's continue. Then after the muscularis mucosa, we have submucosa. Submucosa, it's a dense connective tissue. It's denser than lamina propria. So it's sometimes, by some definitions, submucosa is also claimed as loose connective tissue. But so it's like a transitional stage between dense and loose connective tissue. So actually, okay. So in submucosa we have Meissner's nerve plexus. Meissner's nerve plexus. So this Meissner's nerve plexus, it's usually parasympathetic innervation of digestive tract. So also includes sympathetic innervation. So and actually this Meissner's plexus, and in lamina propria we also have another plexus, so we'll discuss later. So they together they form the gut brain. Sometimes it's named as gut brain, because the interconnection between them is very complicated, and not as complicated as in brain, but still it gives some autonomy to digestive tract. Okay, later it will be discussed in details. But submucosa, so it's it's fibrous tissue, so very dense fibrous tissue, so which makes gut a very powerful, strengthful, and can exert pressure, can exert stress. So same as in the respiratory tract. But in respiratory tract we have next layer. Which is in the digestive tract is muscularis externa. Muscularis externa is a muscular layer, and usually composed of 2 layers. So it has circular and longitudinal layers, but in some position it has oblique layer, for example in stomach. So we also will be discussed later. But predominantly it's inner circular and outer longitudinal layer. And between 2 layers of muscularis externa is present Auerbach's nerve plexus. So that's what I said about. So Auerbach's nerve plexus together with Meissner's plexus and some nerve endings in lamina propria, together they form the innervation of digestive tract. So muscularis externa composed of smooth muscles, with a few exceptions. In esophagus and anal canal, also this muscularis externa from smooth muscle layer converts into skeletal muscle layer. But okay, predominantly it will be smooth muscle layer. Then serosa or serous membrane. It is so serosa. So one details. We have serosa or adventitia, but it is not the same thing. Serosa is the serous membranes. Serous membranes include, there are 3 serous membranes in our body. They are pericardium, peritoneum, and pleura. These three serous membranes. And of course, if we talk about digestive tract, it is covered only with peritoneum. And peritoneal peritoneal lining, so is not for every organ of digestive tract, so because some of them are present in peritoneal cavity and they covered with serosa. But in those areas where digestive tube is not covered with serosa, so it's present retroperitoneally, or like esophagus, it's present in mediastinum. So this instead of serosa we have adventitia. So what is adventitia? We already discussed in respiratory tract topic. So it's okay. It's a connective tissue. So if we talk about serosa, it's a thin layer of connective tissue, which is covered by epithelial lining. So which is mesothelium, lined with mesothelium, and which controls the normal amount of fluid which is present in a peritoneal cavity. And adventitia, it's a pretty thick connective tissue layer, but it's much looser, and that's why it connects different organs between each other. And as I said you before, adventitia doesn't belong to some respectful organ. So it's like media between different organs, so which connects them together. And adventitia is responsible for spreading, serosa and adventitia are responsible for spreading of blood vessels, nerve supplement, and lymph drainage. Somewhere in deepness of this adventitia might present lymph nodes, so which are responsible for immunity response in these organs. Okay, let's talk about mucosa in details. So now I'll show you this is here, but later I'll show you this on diagram. So mucosa is composed of 3 layers. It's epithelium, lamina propria, and muscularis mucosa. So take a look. So which type of epithelium is predominantly present in digestive tract? Of course, it's a simple columnar epithelium. Simple columnar epithelium with a few exceptions. It starts from esophagus. It is lined with a stratified squamous epithelium. In stomach, it's a simple columnar, small descent simple columnar, large descent simple columnar. So that's why you have no. I hope you won't confuse it. So predominantly it's simple columnar epithelia, but in areas like esophagus, oral cavity, esophagus, and pharynx, it is composed of stratified squamous epithelia. Particularly, we discussed it in respiratory tract. And here you see that in esophagus we don't have serosa, and we have adventitia only. But here serosa is like connection, yes. So, and this serosa, 2 leaflets of this serosa forms a very important structure, very interesting and important structure, which is known as mesentery. This is mesentery, and this mesentery connects this digestive tube towards the bones, I mean backbone, or to any other hard structures which maintain their location in digestive tract. So anatomically you discuss the location of digestive tract, so I hope we shouldn't discuss it in details. Glands. So what we should talk about glands in digestive tract. So in digestive tract, so we usually encounter with exocrine glands. Exocrine glands, they are responsible for lubrication first of all, and they are like epithelium, composed of epithelium, and the main cells of main example of the cells, they are goblet cells. Goblet cells, same as in respiratory tract, I remind you. So goblet cells produce mucus, which is necessary for lubrication and moisturizing of surfaces. Then in lamina propria, we also have exocrine glands in lamina propria. It's invaginations of epithelium lining into the lamina propria. And examples of this are gastric glands, which are gastric glands, and also in colon we have these glands, so which are very deeply located in in lamina propria. And sometimes gland deepness reaches the even submucosa. And there are a few examples in our digestive tract. So one of them is Brunner's glands. Brunner's glands, which are located in duodenum. So physiological role of them we'll discuss a little later. These Brunner's glands. Okay, and sometimes glands are located external to digestive tract. So external to digestive tract. In embryology of this digestive system, so we'll discuss it. So how it is arranged. But you already heard about it. It's a liver and pancreas. They actually embryologically they derive same as every of this gland, but they became so outpoached, so independently evolved, so independently organized, so that's why we claim they're distinct organs, but actually they are connected to the digestive tract, and primarily were outpoached from the digestive tract. Then we have endocrine and paracrine cells. They are belonging to the diffuse neuroendocrine system. So a little bit we didn't talk about it. I'm confused. So endocrine, so what's the difference between exocrine and endocrine glands? Exocrine secreted on the surfaces, in the orifices, into the surfaces of epithelial cells, but endocrine and paracrine cells, they produce their secretion into the, and you can say blood, but no, they directly produce it into the connective tissue, so which are present nearby, and then from connective tissue it reaches to the blood. So endocrine glands, you remember, which we discussed in previous chapter. So endocrine glands, they produce secretion not inside the blood. It's not fair to say. It produces first of all in interstitium, and then from interstitium it carries out into the blood. Okay. But these endocrine glands which are present in the digestive tract, they are not. They can be claimed as a distinct organ. It belongs to diffuse neuroendocrine system. It's spread out along the gastrointestinal tract, and it first of all it responsible for secretion of glands, so control the secretion of glands, secretion of gastric juice, for example, is controlled by one of these hormones like gastrin. Okay, and motility of gut and motility of gallbladder, for example, like cholecystokinin, is produced by this endocrine and paracrine cells. Okay, let's move forward. So let's start the discussion about directly organs of digestive tract. So it is esophagus. Esophagus so is lined with the epithelium, which is named stratified squamous non-keratinized epithelium. So I remind you, skin is also lined with stratified squamous epithelium, but it was keratinized. But in esophagus, it's non-keratinized. So I hope you remember it. Same type of epithelia became common in cases of pathology in respiratory tract. You remember, yeah? So in case of hard smokers, yeah, so when chain smokers for 20 years, this normal pseudo-stratified epithelium of respiratory tract is changed to stratified squamous non-keratinized epithelium. And here we can see that it's normal changes in tubular organs. So when it exerts some frictions, exerts some noxious stimuli, so it's a mechanism of safety, is a change this epithelium into the stratified squamous epithelium due to mechanical friction. Okay, then lamina propria. Lamina propria, which is present below it. So here I can LP is shown here. Lamina propria. So it possesses cardiac glands in a cardiac region. So main function of this, of course, secretion of sodium bicarbonate. Sodium bicarbonate, which is released into this area for what? So you can understand. You should know that in a stomach we have high acidic, very highly acidic milieu there, and just for neutralization of acidic milieu, which sometimes might enter into the esophagus due to vomiting or just regurgitation. So we have to neutralize it, and for this reason we have esophageal cardiac glands. So because of cardiac region of stomach, which is present nearby. It's named in name of stomach region. Okay, lamina propria. But in most layers of esophagus, in lamina propria we have no glands, not so evolved glands. Next layer of esophagus is present with submucosa. But in submucosa we have mucus secreting esophageal glands. Submucosa, mucus secreting esophageal glands, which is necessary for lubrication, just for lubrication of esophagus, because the functional role of esophagus is, of course, is just passage of food from the oral cavity and pharynx through the esophagus into the stomach. So because just this function. And a little extends time for digestion digestion by saliva. So you know that in saliva we have some enzymes which also permit its function in a way toward the stomach. It's also kind of necessary. So here once again mucosa, lamina propria, very little lamina propria, and muscularis mucosa. Muscularis mucosa. So have we discussed it here? No, we haven't. I haven't said about it. So muscularis mucosa here is responsible for formation of ruga or some longitudinal wrinkles of esophagus, which allows a one-directional move of the food. This is made by muscularis mucosa. It doesn't necessary for peristaltic movements, but here it's for mainly for formation of the special shape of esophagus. Then submucosa, as I said before, it's mucus secreting glands are present. So this is a duct of mucus secreting gland. Here is the gland itself, and here we have a lot of blood vessels. Same Meissner's plexus is present here, which controls the secretion of this mucus. It nervous Meissner's plexus, and it controls the contraction of muscularis mucosa here to arrange proper shape of the esophageal mucosa. Okay, below the submucosa, it's of course it's connective tissue, dense connective tissue, submucosa, yeah. And then below the submucosa we have muscularis externa. So muscularis externa, it is composed here of two layers, and you see it's made up of inner circular and outer longitudinal layers. It's pretty unusual, so later I'll explain it to you. In upper third of the esophagus, both layers contain only skeletal muscle fibers, on which nerve fibers and cranial nerves 9 and 10 end as motor end plates. So you see, in upper portion, you remember what is upper than esophagus? Of course, it's pharynx. And pharynx for swallowing movements use, of course, skeletal muscle because it should be under our willing control. So same about esophagus. So because we don't have a border line between esophagus and pharynx, so that's why some muscles of pharynx extends to esophagus, and they are supplied by the same nerves, like 9 and 10 nerves, and responsible, of course, for swallowing. And that's why here we have some layer of skeletal muscle fibers, but later it gradually converts into smooth muscle layer, and this situation and until in the end of esophagus, of course, we don't have skeletal muscles, we have only smooth muscle linings. Okay, distally towards the stomach. Adventitia composed of loose connective tissue, of course. Adventitia, so same as everywhere, is composed of loose connective tissue, so trunks of nerves, trunks of blood vessels, which supply it, so they are spreading along the adventitia. So here on this diagram it is not shown. Here it's visible. So this is a detailed picture of mucosa. Once again, stratified squamous non-keratinized epithelia. This is basement membrane. Here below it we have lamina propria. Lamina propria with the little blood vessels, capillaries, which play a role in the formation of inflammation here sometimes. Then below the lamina propria we have muscularis mucosa. Number 6 is it's not shown here. So here is muscularis mucosa, and number 7 here is a submucosa, of course. Okay, back. So later below the muscularis mucosa, so I hope you understood. Yes. So here below the muscularis mucosa we have submucosa. Here we have mucous glands, and below the muscularis mucosa, below the submucosa we have circularly arranged smooth muscles and longitudinally arranged smooth muscles. In upper portions it is skeletal muscle, of course. Okay, so here it's shown. So for example, this is typical skeletal muscle picture. So you see that in some portions of the esophagus we have skeletal muscles. Here we have smooth muscle. This is orbax plexus. So it gradually converts from skeletal to smooth muscles, in distal portion especially. So here you see this pharyngeal muscles, and from pharyngeal muscles it extends here. So this is typical muscles, but which are included in a wall of esophagus. Now let's talk about our stomach. Of course, stomach. So the first portion which encounters with the food is known as cardia, and you remember we discussed cardiac glands of esophagus. They are and here in cardiac region of stomach we have the same glands, cardiac glands of the stomach, and main action of this cardia, of course, is secretion of mucus containing sodium bicarbonate, just to prevent acid aggression to this area of the esophagus. So it's a safety, it facilitates safety of this esophagus. Okay, and then we have fundus and body. So functionally fundus is the most active secretory portion which produces most of HCl and enzymes of the stomach, so like pepsin, of course, which are produced predominantly in fundus. Almost similar function present in body, but a little more scattered function. And of course we have pylorus. So pylorus is like in cardia. In pylorus we have pyloric mucus-secreting glands. This pyloric mucus-secreting glands serves to the same function, absolutely similar function. It serves to prevent aggression of acid, but not to esophagus like in cardia, but to duodenum, which is following here. You already know anatomy in physiology, I guess you understand that acidic milieu of stomach is aggressive for both esophagus and for duodenum. And this cardiac region and pyloric region so is distinct from rest of the stomach just because of their possession of these mucus glands with the opportunity to eliminate aggressiveness of gastric juice. Then fundus by its active secretory role. So let's talk about structure of the stomach. Structure of the stomach, it contains surface epithelium, so which is a simple columnar epithelium facing the lumen is modified so that all cells secrete mucus, forming a sheet gland that protects the stomach from its acidic environment. So surface epithelium, so in details I'll show you a little later. The surface epithelium, and this surface epithelium forms the projections, and cells of surface epithelium formed in projections. This projection is known as gastric pit. This gastric pit is actually it's a duct of gland of stomach. So, and below this gastric pit we have actually gland of the stomach. Stomach glands, they are very abundant in stomach, and here on this diagram it's visible. This is a normal epithelial lining of stomach, and this lining continues here in gastric pit because it looks like gland. Really, it looks like gland, but actually it is just continuation of normal epithelial surface, just to prevent aggressiveness of secreted here in gland products. Okay, so gastric glands. Gastric glands, they are simple branched tubular glands, begin at the gastric pit and extend through the lamina propria to the muscularis mucosa. So as it's shown here. So here we have muscularis mucosa. Between these glands we have a very little amount of loose connective tissue which form these glands actually. So region of glands that attach to the gastric pit is called the neck region, the base region, or isthmus. The base region of the gland is located adjacent to the muscularis mucosa, as I said before. So secretory cells in these glands vary in each region of the stomach. So here we see different secretory cells, which are like parietal cells, enteroendocrine cells, and of course chief cells. And about them we'll discuss a little later. So also here present mucous neck cells. Okay, a little later. No, right now. Yeah, I forgot my lecture. So here you see, this is the structure of gastric gland. So in every region it varies in their consistency, and we will discuss it now. What's the difference? But general plan of this gastric gland is here. Once again, surface. So here we have an gastric pit, which is covered by surface mucous cells. Later, so in the neck region of this gland, we have mucous neck cells. They are like same as surface mucous cells, but they are more extensively producing mucus. More extensively, just because here very aggressive juice coming. Okay. Later, behind it, we have scattered parietal cells. So these parietal cells, take a look on the structure of these parietal cells. It's very specific. Inside the cell, we have a duct. So it's a very strange situation. Usually we have duct which is lined with epithelial cells, but here we have duct inside the cell. It serves for increasing of surface area. Increasing of surface area of the cells to produce more HCl. You see here, and also organelles which are present here in parietal cells, they are mitochondria. They are mitochondria which produce. So you remember one of function of mitochondria from histology of first semester. That one of function of mitochondria is it's a transportation of H plus ions so on the surface of through the membrane of mitochondria, and then these H pluses are used in process of joining with oxygen in respiration, and then this process, when H pluses which are produced by mitochondria joined with oxygen, so it leads to formation of ATP itself. Yeah. So it happens after Krebs cycle, because in Krebs cycle there are no ATP produced, yeah, but it is produced by formation of H pluses, and these H pluses so protons of hydrogen ions which are produced here, they are joined with oxygen and then forms the ATP. But here, these H pluses, they go not to formation of ATP, but they go outside, for excretion, into the gland, inside this gland. And of course, following to the H pluses go the chlorine anions, because we can produce just positively charged ions without following of negatively charged anions. So okay, they are secreted in this gland, and of course, so gradually by diffusion it enters into the stomach. So that's how, because of these few parietal cells, you see there are not too much parietal cells along the digestive tract, but these parietal cells produce a lot of hydrochloric acid. So which allows to, so might be you remember, so pH of gastric juice is approximately 2 equals to 2, so it means 10 in power minus 2 concentration of H plus ions. So this is it is made just because of the parietal cells, and most actively it is present in fundus region of the stomach. Okay, it also here one more additional function. It's intrinsic factor, which is produced by parietal cells. This intrinsic factor is necessary to join extrinsic factor. Extrinsic factor which is B12, and only together with this intrinsic factor joined to extrinsic factor, when it enters into small intestine, it can be absorbed by small intestine. So without intrinsic factor, B12 vitamin cannot be absorbed in digestive tract. That's why some pathology of these glands, which we'll discuss in following topics. So it leads to B12 deficiency anemia. Also, we'll discuss it in next topics. Then next, chief cells. So name chief means they are holding the main function here. The chief cells they producing, of course, pepsin enzymes. They secrete pepsin, but here it's named pepsinogen. So what's the difference between pepsin and pepsinogen? Of course, pepsinogen is an active form of enzyme. You discussed it in physiology. And pepsin, it converts into pepsin only in a hollow space of the digestive tract. So because pepsin is very aggressive against proteins, and each of the cells which are shown here, they are proteins. Yeah, they contain proteins, and cell membranes contain proteins, and they can be preserved against it. And that's why we can activate these enzymes only in orifice of the stomach, but not inside the glands. Then we have so gastric lipase in less amounts are produced.
Sir, your voice is not clear, sir. It's cutting.
Now, is it clear now? So a little technical issue. So let's continue. So chief cells, they produce pepsinogen and pepsin. So these enzymes which cause the protein degradation, protein hydrolysis. That's why it's named as hydrolytic enzymes. Pepsin, but it's present only in orifice of the stomach. It's activated in orifice of the stomach, but produced here in gastric glands, in the deepness of this gastric gland. And also here we have a lot of G cells. G cells which producing gastrin. So this gastrin which stimulates the synthesis of parietal cells and chief cells. So they respond to extension power of stomach. So when stomach is too extended by food, gas, or any other, might be by tumor. These G cells produce a lot of gastrin in activation of parietal cells and chief cells, and also produce a lot of mucus. So gastrin is like stimulator cells which responds to stretching of the stomach. So that's why please keep it in mind. So when we in pathology especially, it plays very important role. So but main function, first function, of course, so it's logical understandable function. So when we don't eat, so our stomach is empty, so it is not stretched, then there is no sense to produce gastrin, and this gastrin is not produced. But if stomach is fulfilled with something, with some material, with I don't know, might be with food predominantly. So these G cells producing gastrin, and in aim to produce more acidic milieu, and of course pepsinogen for protein destruction. Okay, you had physiology of GIT, or if you didn't, so it will be very soon. So then it is explained what the reason of it. Okay. Of course, below it we have same lamina propria. So in between these glands we have lamina propria. Below it we have muscularis mucosa. Then after muscularis mucosa we have some mucosa, and so on. So muscularis externa, which we'll discuss soon. And of course around it we have of course serosa. So serosa because stomach is intraperitoneal organ, mesoperitoneal, but it doesn't matter. So muscularis externa. Muscularis externa of stomach is composed of internal oblique layer, middle circular layer, and outer longitudinal layer. So please remember this. Step by step. Yeah. Internally we have oblique layer. So actually, internal oblique layer is a just a special type of circular layer. So it grows grown from the circular layer, but it's little change the direction. That forms in internal oblique layer, a middle circular layer, and outer longitudinal layer. So in between this middle circular layer and longitudinal layer, we have Auerbach's plexus. So it's pretty understandable. So if we look on this like one layer and second layer, yeah. So like internal circular layer and outer longitudinal layer. Of course, as everywhere in the digestive tract, between this outer and internal layers of muscles, we have Auerbach's plexus. But internal oblique layer is outpoached from middle circular layer. But they sometimes interdigitate, and as written here, making difficult to distinguish one layer from another. But we have no actually clue with this, so we don't have to do this. Okay. This muscularis externa, you know that it's functionally, so here it's shown, yes, this muscularis externa pretty thick, and it's hard to distinguish, so this oblique layer or circular layer. And this is Auerbach's plexus, obviously present here, so this connective tissue with nerve endings here, with nerve fibers, and this is externa, muscularis externa, it's outer longitudinal layer. What can we say here? It replies with the function of the stomach. So, what the function of stomach? Of course, it's not only formation. Is not only storage of food. It's not only for creating acidity to prevent bacterial invasion. Yeah, because it suppresses the bacterial invasion, and it's not only for portionate transport of food there, but also it's for mixture of food with the gastric juice. So digestive function of stomach is made not only by secretion of this gastric juice, but also by intermixing of this gastric juice with the contents of the stomach. So and that's function of muscularis externa here. So variations specific to the cardiac region. So in cardiac region, I remind you, cardiac region is which is present very closely to esophagus. It contains abrupt transition of epithelium from stratified squamous moist of the esophagus to a sheet gland lining the cardiac stomach. So here, this is example. You see, very abruptly. Here we had stratified squamous epithelium, and here simple columnar. So here it's hardly visible, but if we'll make a zoom here, so you can see this simple columnar epithelia. But you see, epithelial lining is absolutely different with the glands. Yeah. Okay. Then length of gastric pits is about equal to the length of cardiac glands. So gastric pits are very long. Yeah. So here is the gastric pit, and below this gastric gland. They are very long because of its aim is to prevent aggression to this stomach juice to the glands. Then cardiac glands primarily secrete mucus. Also other products are also produced. Glands are frequently coiled. Okay, cardiac glands so primarily secrete mucus. So of course to decrease the acidic milieu, to neutralize the acidic milieu in this area, to prevent aggression to the esophagus. Cardiac glands of the stomach extends into the lower esophagus, forming the esophageal cardiac glands, as we discussed before. Fundiglands. Fundiglands are about twice as long as their gastric pits. So they're much longer. They're more effective. So which cell types present there? Like stem cells, mucous neck cells, parietal cells, same by function. Chief cells, and enteroendocrine cells, same cells as we discussed before. So and they contain maximum amount of chief cells and parietal cells. Yeah, here a specific type, or so actually not specific, but they're pronounced. They are stem cells from which different types of epithelial cells are present. They are, because here a very aggressive meal is produced, a lot of cells suddenly died. Yeah, and then we need some cells to replace them, and they are located in neck region, and they gradually replace these dead cells. So these chief cells and so parietal cells and chief cells are present. So you see, if you see a lot of mitochondria, oxyphilic cytoplasm, and some extension which are known as intracellular canaliculus or intracellular ducts, you can easily say it's parietal cell. But in a chief cells we can see secretory granules with the enzymes. So here, this cell will be mixed. It will be very acidophilic due to presence of proteins here in granules, and but cytoplasm will be very basophilic because of roughened plasma reticulum presence here. And I remind you that roughened plasma reticulum contained ribosomes, and these ribosomes are containing RNA, rRNA. And this rRNA, of course, attaches so basophilic stains. Because it's acid, of course, it attaches basic stains. So proteins predominantly attach oxyphilic stains. I mean, using glands. So mechanism of HCl secretion is somehow shown here. So, but you can skip it. You can use just little explanations that mitochondria with this energy splits the molecule of water into two portions, and HCO3 is produced outside, Cl and H pluses produced outside. So HCO3 is together joins with sodium outside, and chlorine joins with H pluses and produce HCl here. So okay, I don't agree with these diagrams in some portions, but okay, doesn't matter. Variation specific to the pyloric region. In pyloric region, pits are longer in pylorus than in the cardiac region. So you see, pits are very long. So because we don't have to save themselves with the with the juice which is produced there. Pyloric glands not as coiled as in cardiac region, primarily secrete mucus, so you can skip it, actually not so important. And then enteroendocrine cells are also present here. So as everywhere, a circular layer of muscularis externa is greatly thickened to form the pyloric sphincter. So yeah, muscularis externa. So muscularis externa. So yeah, here we have diagram, I guess. Yeah. So here we have a diagram in small intestine. So muscularis externa in some portions get very thick. So not here. Sorry. I'll find you. I had this pyloric sphincter. I excluded this diagram from this lecture. Sorry. But pyloric sphincter is a smooth muscle. Very thick smooth muscle, so which don't allow the all stomach contents to enter in the duodenum, because amount of glands which can be amount of mucus which can protect the duodenum is limited, and of course it produces it gradually, and also for proper digestion we have to give a portionate amount of the food from the stomach into the duodenum. Okay, physiological role, you know. So summary you can read by yourself, so I will send this PPT to you. Then small intestine. So what can we say about small intestine? Structure that increase the surface area of small intestine. So okay, first of all, small intestine is the main area where digestion happens in self. For example, in stomach, absorption of food does not happen. So only a few compounds like a little bit of water, little bit glucose, and alcohol can be absorbed by stomach. Actually, alcohol can absorb even in oral cavity, but and esophagus also. But in stomach, it is a little more abundantly present. But in small intestine, all the products can be absorbed. For this, we have special cells. and entire apparatus of small intestine is aligned for absorption. And for this we have microvilli. So it's a microvilli is a structure of the cell. So you see each cell contains projections of their cytoskeleton, which form as a brush on the surface of cells. And it is named as striated border. So we see one cell contains microvilli. Okay, but then villi. Then we have villi. Villi is a structure which is made by the cells. So there are many cells forming villi. You see, microvilli is a property of one cell. Villi is a property of a few cells. And actually, it's a protrusion of lamina propria and overlying epithelium into the lumen. You see, it's very big difference because a lot of students. So every year I encounter with the same feature that students used to confuse microvilli and villi, and they don't see difference. Microvilli is like small villi. No. If you answer like this, so I can directly put 2 marks to you in practical class. And if it in case of offline exam, so I will put you 2 marks to the answer. Microvilli is a small villi. No. Microvilli is a projection of cytoplasm into the orifice, but villi is a projection of lamina propria. So you see the difference. Yeah, lamina propria is a tissue. Microvilli cytoskeleton is a portion of the cell. Yeah. So okay, on diagram might be it will be visible. So and villi assume different shapes in each of 3 intestinal subdivisions. So you see lacteals are present inside this villi. So what is lacteal? So actually lacteal is a duct of lymph vessel. It's a lymph vessel duct, so which actively absorbs fluids from small intestine into the digestive region, into the lymphatic system of small intestine. And then individual smooth muscle cells lie parallel to the long axis of each villus, milking the lacteal contents of the periphery. So you see these smooth muscles present inside the villi, just in a goal to direct this lacteal, direct this lymphatics towards the center of towards the centers of lymph nodes. Then we have plica circularis. Plica circularis is a structure of projection of submucosa. You see, it's a next level, yeah. So next level of increase in the surface area. So plica circularis is a projection of submucosa, which causes on which present villi. Okay, let's talk about it on diagram. So this is, for example, plica circularis. You see plica circularis. They are visible with naked eye. So you see it's projection of submucosa on the surface of which we have villi, and on the surface of villi we have microvilli. So here is the diagram. This is villi. You see, it contains a lot of cells. Inside we have lamina propria, in lamina propria we have blood vessels. This is artery which supplies it, and backward goes vein. Floating these back. Then and here blindly started and continues deeper. It's known as lacteal. Lacteal lymph. It's a lymph capillary which is present in each of these villi. So actually this lacteal is named because of milk. So lactum, it's a milk. So lacteal, it's a whitish fluid because of what? Actually, its main goal is absorption of fatty products which are present in small intestine. And so you know that actually it's not directly swallowing here, but it is explained by the cells which are present in mucosa. So let's discuss it. Mucosa of small intestine is composed of 2 types of cells, mainly absorptive cells and little bit goblet cells. So absorptive cells, there are cells which main function is absorption of food from the orifice. So here you see, this is a cell, one cell, and on the surface of each cell we have microvilli. So sorry, this is microvilli actually. this microvilli which absorb from the surface of intracellular digestion happens with them. So and here all the products like carbohydrates, amino acids, oligopeptides, fats, which are dissolved in the small intestine, they are absorbed by microvilli into the into the cytoplasm. So what happens in cytoplasm? In cytoplasm, final digestion happens. So here we have a lot of enzymes. We have some lysosomes which destroy oligopeptides into the amino acids, which small chains of carbohydrates change into the glucose, fructose, maltose, or any other monosaccharides, and travels it into the lamina propria. So here, this direction happens here from the orifice towards the lamina propria, and all the products which are absorbed, they are thrown into the lamina propria. And in lamina propria we have capillaries and lymph, blood capillaries and lymph capillaries. Yeah, and lymph capillaries is lacteal. Blood capillaries is blood capillaria. Okay, and what's the pathway of fats which are absorbed in digestive tract? So fats, they have little complicated pathway. So they are absorbed here. Here in a, so you see we can digest different types of fats. So they are, some of them are useful for us, some of them are useless. And that's why we have mechanism by which these fats, all of them are getting hydrolyzed. They getting absolutely dissolved in a small portions, but they're reconstructed again. They're reconstructed again with the help of smooth endoplasmic reticulum. And here also we have rough endoplasmic reticulum. So you can see, so what's the reason? It's absorptive cell, so it shouldn't produce proteins. It should just transport food from intestine to the blood. But actually, proteins, so fats, as you remember, they are water insoluble, and we can just direct them into the blood or lymph node, into the lymph. So we have to envelop it in something water soluble, and the best envelope for the lipids is proteins, of course. And proteins which are produced on rough endoplasmic reticulum, which is present here, yes, so it's marked here. It forms the apoprotein. It forms apoprotein which envelops this fat, not only fat but lipids, any lipid which is present, envelop it, and then this coordination of fat and proteins is named as chylomicrons. It's a transport form of lipids, and then these chylomicrons transported into the interstitium, and from interstitium they are pretty large molecules. Yeah. If we talk about glucose and amino acids, they are very well water soluble, and they can easily be absorbed by capillary. But here we have large molecules. They cannot transport into the capillaries. They can be transported only into blind-ended lymphatic system, and that's why these small chylomicrons they give this white color to the lacteal, white color to the lymph capillaries which are present here. So also here we have goblet cells. Functional goblet cells remain same, producing mucus, and this mucus which is produced, so in one hand it saves the surface from physical and chemical friction, and secondly, of course, it necessary for moisturizing and smearing of the surface of small intestine with saliva-like secretion, with the mucus. Yeah. Intestinal glands. So in intestine we have intestinal glands. They are known as crypts of Lieberkühn. So 1 minute, so time. Okay, crypts of Libercun, they are simple tubular glands that begin at the basis of the villi in the mucosa and extend through the lamina propria to the muscularis mucosa. It possesses different cells. They are absorptive cells, goblet cells, Paneth cells, enteroendocrine cells, and M cells. So before it, we discussed only about absorptive cells and goblet cells. So let's talk about Paneth cells. So here they are Paneth cells. So Paneth cells, they are just containing some eosinophilic inclusions. And actually nowadays we don't, so here it's shown much more detailed. So this is, you see, villi of small intestine. Here starts the, here the gland starts. Here gland starts, and the crypt of Libercun. But in duodenum, it forms the Brunner's gland, so we'll discuss a little later. But here, in crypt of Libercun, we can see these Paneth cells. These Paneth cells, we are not confident what is it. So some of them think that it's secretory granules, which somehow facilitate the activity of cells nearby. But others are pretty sure that this is cell membrane, cell walls of bacteria, which are normally present in the small intestine, which ingested them and save this cell walls for analyzing of it and producing of antibodies against them or any other cellular response to this. So any immunological response to these cell membranes, cell walls. So okay, next I want to talk to next is enteroendocrine cells. So enteroendocrine cells, here they produce similar, so they are here this enteroendocrine cells are present. No, it's M cells. Yeah, they are scattered here. It's hardly to find it. So here it is, for example. Just imagine this is a cell. Enteroendocrine cell is responsible for secretion of cholecystokinin and also gastrin. Cholecystokinin is responsible for gallbladder constriction, smooth muscle constriction of gallbladder to release this, to release the bile from the gallbladder into the duodenum. I hope physiologically you remember it. So and later, and also gastrin, which gives a signal to the stomach, possibly so if it's overfilled, so to give signal to the stomach to produce more gastrin, to produce more enzymes, pepsinogen and HCl. So might be might be also present here. And then finally, most interesting, it is M cell. M cell is an empty cell which with the presence of lymphocytes inside and macrophages. So you see one cell inside contains lymphocytes and macrophages, and also here enters the dendritic cell. So actually this cell play a role like thin cover against immunity system to the small intestine. So in small intestine, again I repeat, we have a very huge microflora. This microflora contains a lot of bacteria, so which in some cases it assists to our digestion, but in some cases it can cause danger. It causes potential danger for us. For example, you know, when we die, so the process of decomposition in humans. So why actually corpses smell so badly? So it's because of this bacteria of small intestine and large intestine, both. This bacteria evolved very hardly, and only immunity saves them from, saves us against their aggression. Immunity so play very dangerous game with them, yeah. And these bacteria which are present in small intestine, they after death of our immune system, they will digest our bowel, yeah. So that's why you can. It's usually in most of conferences they say microflora of our small intestine is the best thing which we can imagine. But actually, directly after death, it betrays us, and they use our body as just as a food. Okay, and this is necessary for antigen presenting of these bacteria. These M cells, they just capture some of this bacteria, make some probes of this bacteria, and lymphocytes which are present here, they analyze it, give the signal to macrophages, to dendritic cells, to make it in charge, to be in charge. So what's the composition of bacteria here? If any aggression from this side, from the orifice of small intestine happens, so we have to react on this very fast, very fast. Okay, so do we have a little time? Actually, 1 hour of lecture passed. In beginning also we discussed something. Okay, that's enough for now. We'll continue topic about duodenum in next class.
Okay, sir.
Thank you for attention. This lecture will be uploaded into the YouTube, into the same playlist on YouTube. So now I'm checking your attendance. Okay. So don't forget to write your name in the chat. Okay, it's your confirmation that you are here. Okay, thank you. See you next time. Goodbye.